So thanks a lot. It's really neat to be here, and this is actually one of the first investor conferences that we've been to. So we're really trying to make an effort and to try to get investor funding. My name is Adam Robertson, and I am the Chief Scientific Officer at Hemispherian Oncology. At Hemispherian Oncology, we're advancing two pharmaceuticals for the treatment of Glioblastoma Multiforme. Glioblastoma Multiforme is a rare, deadly disease that tends to affect, it's the 17th leading cause of cancer in adults, and among children it's particularly bad. It's the second leading cause of cancer in children. In addition, it's the 7th leading cause of death in children. In the Western world, and particularly the EU, it's the second leading cause of death. At the same time, we're spending a lot of money for this particular indication. We spend about $72,000 per patient per year on this indication. And in stark contrast to many other types of cancers, which have significant survival rates, and survival rates are improving, brain cancers and glioblastoma multiforme in particular are not improving, have not been improving, and are relatively low at about 1%. Now you can look at the treatment options, and there's just not a whole lot out there for the treatment of glioblastoma multiforme. There's surgical resection. 48% of patients are eligible for surgical resection. 52% are not eligible because they would not be expected to survive the surgery. There's also radiotherapy, and if you're treated with radiotherapy, after diagnosis you'll survive for 12.1 months. The frontline therapeutic, if we look at chemotherapeutic molecules, there's one frontline therapy and no generally accepted second-line therapy. Temozolomide, the frontline drug, if you're treated with it, you will survive an extra 2.5 months longer. And this is also a drug that made Merck, when it was on patent, about $1 billion a year. Glia Dell is another drug that's typically indicated for this, for if you happen to have surgery, and this drug must be placed directly into your brain. It only works if it's because it doesn't pass the blood-brain barrier. It only works if it's placed directly in the brain, and this will allow you to survive for another 1.8 months to 13.9 months. And this is making about $20 million a year. For us, while this is very bad for the patient, this is a pretty good opportunity. The bar for success at this point for a successful drug is very, very low, 2.5 months. So improving survival to 2.6 months would result in a $1 billion a year drug. So then, of course, how did we end up stumbling onto a glioblastoma multiforme drug? I ran an academic laboratory. I was a group leader at the University of Oslo for about 10 years. So we worked on and off on this project for a significant amount of time, and we were very interested in a new DNA pathway by which the cell regulated licensing. And licensing is a way in which an organism or a cell ensures that its DNA is replicated one time and only one time. Now, unfortunately, I don't have time to go into the details of this entire pathway, but it is published down here. And that's, if you're interested, I can talk to you about this later, or if you'd like to read the publication as well. But I think the interesting part about this is how we were able to target this. One of my PhD students decided to target this mechanism, and she kind of thought that because this mechanism is present in all cells, but it's only targetable in cancer cells because in normal cells it's saturated. And so we can very effectively target cancer cells, but not target normal cells. And this was kind of our first indication that this mechanism of action was targetable by these replication licensing inhibitors. We call in these molecules GLIX1 and GLIX5, and we can see that this is a brain tumor from a 13-year-old boy that eventually did die from this glioblastoma. And if we move from left to right, we can see that if we treat with temozolomide, this is completely resistant to temozolomide. And this is like 74% of glioblastoma tumors. They're completely resistant to the frontline treatment. If we treat with GLIX1 or if we treat with GLIX5, we see that we're able to effectively kill this glioblastoma. So we think that this patient may have had a better outcome if we were able to advance our pharmaceuticals a little bit more quickly. Of course, this is just one of our first indication that it would work and one of the first assays. And we're going to skip a few steps here, but now we jumped into mouse xenograft assays. In this case, we had 24 mice, and we had 24 tumors. We implanted these tumors into the mice, and then when the tumors reached a predetermined size, the mice were randomized. and set into 3 different groups of 8 mice. In the control group, we can see that these tumors grew relatively large, and in one case, the mouse actually died. However, we don't think that this was the result of the tumor, just some other effect. If we see GLIX1, we see regression in 8 out of 8 tumors, and 4 out of 8 were regressed to the point there was no tumor at all. We see a similar effect with GLIX1. We see regression in 8 out of 8 tumors. So I think these results are pretty compelling, and they suggest that we do get good efficacy with these molecules. But it's important to note that we didn't need this good of a result. To make money with the glioblastoma multiforme, you need to have survival better than 2.5 months. I think these results suggest that we can do much better than 2.5 months, and therefore we can make a lot of money doing this. In addition, we had this verified at the National Institutes of Health. I ended up running into the chief of neurosurgery at the National Institutes of Health. I showed him some of this data that we had. He liked the idea, and he ended up getting one of his scientists to try to reproduce some of this in his model system. He was able to see cytotoxicity in all the cell lines he assayed, and these are patient-derived neurospheres, which he tells me are the closest to treating a glioblastoma in a patient, aside from actually treating a glioblastoma in a patient. So now we can demonstrate that we can kill these tumors in 3 independent labs in 3 countries. So I'm reasonably convinced of the success that these molecules will be successful in the clinic. In addition, we chose glioblastoma multiforme because we had the best efficacy and the bar for success is relatively low. And it's also, so we can see that GLIX5 kills glioblastoma, and it's a rather broad spectrum drug and kills many different types of tumor. At the same time, GLIX1, much more narrow focus, only glioblastoma, and it kills a certain type of leukemia. If we move over here to glioblastomas, we can kill about half the glioblastomas that we treat, and this is a different subset of glioblastomas than would be killed by temozolomide, the frontline drug. Therefore, in combination with temozolomide, we can treat a majority of tumors that would likely have negative consequences for the patient. In addition, we also tested normal human cell lines, and we can show that these drugs, GLIX1 and GLIX5, do not have significant negative cytotoxicity effects in normal human cells, suggesting that these would be well tolerated by humans and that they would be able to, that they would not have significant side effects. And this is also kind of additionally backed up by some of the mouse data that we have. So we can kill the tumor, and we don't have any negative effects for the patient. Another important thing that we can show is that these molecules do pass the blood-brain barrier. They're small molecules, and much like temozolomide, they're very effectively able to cross the blood-brain barrier. This is in stark contrast to antibodies and peptides, which don't pass the blood-brain barrier very effectively. You may be aware that there's several recent clinical trials that failed in phase 3 for glioblastoma multiforme. These were like, most of the time this is antibodies, and the failure was because they couldn't get to the right biological compartment. We can demonstrate pretty clearly that our drugs will get to the right biological compartment to inhibit replication licensing. This is the team, myself and Teresa. We have both left our academic jobs to pursue this drug. We have partnered with Xenu, who is helping run the day-to-day operations of Hemispherian. And we have 2 other scientists, Niels and Kristen, who are helping to advance these drugs towards the clinic. We're partnering with many different companies, but notably the National Institutes of Health. Here in the UK, we're sourcing our IND studies and mouse studies to Charles River. We do our patenting with Danes here in the UK. As far as patenting goes, we have 2 patents. We have GLIX1 and GLIX5 and their derivatives patented as first medical indication. This was filed last year, so we have about 19 years of patent lifetime left on this patent. In addition, we also have a synthesis method for the synthesis of GLIX1. This was filed late last year, so we have almost 20 years of exclusivity for this particular patent. If we look at the market, in the 8 major markets, there's about 50,000 glioblastoma cases every year. And if we compare that to the price commanded for a similar, or a rather not similar, but a anti-cancer drug of $26,000 a year, we can estimate that once we get to the market size will be about $1.3 billion every year. As far as the risk-adjusted net present value for the company, we can see that according to the models and the relevant parameters are entered in, we can see that the company is worth about $10.8 million. And we're trying to advance this now to phase 1 trials with our next fundraising round to triple the value of the company to $30 million. And this is where we need investment to triple the value of the company to $30 million. And that's what we're looking for. We're looking for $1.8 million to scale up production of our drug, file for rare disease protection, and to complete an IND package. We expect that we'll be able to get that completed within 12 to 18 months. We would then have to go back for another fundraising round. And this is the cost breakdown. We need IND studies, which must be outsourced for us. We also need to have salaries for the team. We need to have laboratory and office space and the synthesis of the drug. Altogether, we calculated this is about just under $1.8 million. We presently have funding from 3 different sources. The founders have contributed $350,000 to make this possible. We also have some soft funding from the Research Council of Norway and Innovation Norway. And so we're really happy with the way things are going, but the ability for us to continue funding this, we require more money than we can earn at doing other things. So we need to have an investor commitment at this point. So that's what we're here to do. If you have any questions or you'd like to discuss this, please let me know. Here's my contact information. And any questions, I'd be happy to hear them.